Hello everyone. In this presentation, I am going to talk about one of my recent projects during my PhD at University of Vienna under the guidance of Professor Ulrich Technau. In, in this project, we studied a transcription factor called brachyury, which was identified way back in 1927 while they were studying the effect of radiation in mice. It is a very crucial transcription factor which was later identified to have very crucial function in the mesoderm development in vertebrates. Mesoderm is a middle germ layer which further gives rise to tissues such as muscles, bones and notochord. So very crucial function. Uh, it is also a founding member of the T-box family of transcription factor, members of which also has a crucial function in developmental process. The name brachyury itself means short tail in Greek. It is named such because the heterozygous mutant mouse were found to have short, slightly kinked tail, while homozygous mutant did not survive. So in vertebrate, it has also another function which was recently identified, as it is a marker gene for a group of cells which have a bipotent potential where they can either go towards mesodermal fate or towards the neuronal fate, and brachyury is an important marker of these cells. These cells are called neuromesodermal progenitor cells. So, outside of vertebrates, brachyury, although has been shown to express even in fungi, its function there is not very well studied. So, therefore, in this study, we present a genome-wide brachyury target comparison across strategically selected metazoan species. The goal of the study is to identify crucial differences between triploblastic and diploblastic animals. So, for example, animals wherein there is no mesoderm and its derivatives like bones and muscles and yet show expression of brachyury. So, to identify why there is a difference between the brachyury mode of action, and to also to identify the exact role of brachyury in these diploblastic animals where the canonical role is not performed. To achieve our goal, we identified brachyury targets in strategically selected species wherein we developed antibodies in our model organism Nematostella vectensis, a diploblastic animal where mesoderm is absent. Our collaborators in Italy worked similarly in sea urchin and developed antibodies for brachyury in sea urchin. A ChIP-seq analysis was performed on both of these, and subsequently we also used public data sets for Capsaspora, Ciona intestinalis, Xenopus tropicalis and Mus musculus. With this selection of species, we cover a wide range of species ranging from single-cell eukaryotes to mammals. The effect of brachyury binding was studied with knockdown or knockout experiments with RNA-seq. We made few changes in the canonical approach to ChIP-seq analysis. First, normally a closest gene to brachyury or transcription factor binding is considered as target of transcription factor binding. However, then may not reflect a true biology. So, to improve the target identification, we made use of ortholog information. For this, we needed reliable set of orthologs, and for which we used OMA algorithm. With this usage of ortholog information, there was an improvement in target identification. So, the second change we made in our motif analysis, wherein normally there is an interest in centrally enriched motif. In our case, brachyury is a dimer, so its binding site is palindromic. However, it wasn't known whether the full palindrome is required for activity or the half palindrome is sufficient. In all our data set, we find both half as well as full palindrome. So concluding that both can be functional. Apart from the centrally enriched motif, we also looked for other motifs in the brachyury peak region in hopes to identify candidate or putative cofactors or collaborators of brachyury within the peak region. So those are shown in figure number 3 here. So with this we move to the results. In our analysis, we find some key findings where brachyury regulates itself as it's the target of itself in all species we studied. Key brachyury targets that enforce the mesodermal fate on developing cells are missing in diploblastic animals, for example TBX6, mesogenin 1, MESP1, etc. Brachyury regulates neuromesodermal progenitors even before bilateral lineage. There is a conserved feedback loop involving brachyury, Wnt genes, and FOXA in all animals we studied. And there are certain transcription factor motifs frequently occurring in the vicinity of brachyury, which indicates possible cofactors or collaborative mode of action of brachyury. We now take a look at conserved feedback loop in more detail. Although the conserved feedback loop is there in all animals we studies involving Wnt genes, brachyury and FOXA, it performs variable function in these species. For instance, in Nematostella, it downregulates the neural fate genes and activates the axis patterning genes. Whereas in sea urchin, it suppresses the mesodermal genes while also activating the axis patterning gene and some function in anterior neural fate determination in sea urchin. In mouse, the function is very different, wherein it suppresses neural fate genes, activates the mesodermal gene, and plays some part in axis patterning as well. So, although there is a conserved feedback loop, the function is different in different animals. Here, I would like to thank my guide, Professor Ulrich Technau, to provide me opportunity to work at University of Vienna, and I would also like to thank FWF for the funding. Thank you very much.